So we've discussed aerobic respiration. So that's what we do. We basically have oxygen around. We then go ahead and are able to do glycolysis, the prep, Krebs cycle, and then the electron transport chain. But smaller organisms can't do all those steps. They don't have the luxury of doing them. Or imagine us when we're not around oxygen. We do this other type of respiration called anaerobic respiration. So bacteria do this, we do this, and this is going to be without oxygen. We call it fermentation. It's called fermentation even in us when we don't have oxygen around, but fermentation is basically respiration without oxygen. There's two different types. There's lactic acid fermentation and alcohol fermentation. We'll talk about both and try to figure out which one we do. Anaerobic respiration, just as a recap, basically occurs in the mitochondria and needs O2 present. Anaerobic respiration, and anytime you see this part in front of a word in biology, it means without. So, and then we see aero, that's oxygen, and then bic means to do something. So this is without oxygen. Respiration occurs in the cytoplasm. So basically it's going to be like glycolysis. It is with O2 absent, or it's really limited, and there is no final electron acceptor. Do you remember what the final electron acceptor was with aerobic respiration? It was O2. And remember how the O2 had to come in with the two hydrogens and become H2O? And that was going to be the final electron acceptor. There's no recycling of coenzymes that are going on here. In this version, aerobic respiration, those NADs will pick up the NADH, and then as soon as they drop off those hydrogen electrons to the electron transport chain, they just went all the way back to either the Krebs cycle or the prep stage or glycolysis, and picked up more and just kept going back and forth. Here we don't really recycle them, and glycolysis is the last step. So this is the only thing we're going to do is glycolysis. So anaerobic respiration, there's many different types of anaerobic respiration that occurs. A little bit of ATP is produced, but there's really no final electron acceptor. It might be that some things are going to use something inorganic to be a final electron acceptor, but that's it. And anaerobic respiration is without the electron transport chain, of course, so that means they produce very little ATP. And when carbon dioxide is used as an electron acceptor, the product is kind of methane or acetyl acid. So it depends on the organism. So they could make acetyl acid, it could make, acetic acid, or it could make methane. And as you know, with larger organisms like us or cows, methane is going to be produced. So here are the two processes. So keep in mind this one looks a lot more simpler because these are just one-step sort of issues. They don't do the Krebs, they don't do all that crazy stuff. So as you can see, we have two versions. We have the lactic acid path, and then we have the ethanol path. Same sort of thing is going on, where glucose gets split in half. With both of these, you can see glucose just gets split in half. With both of these, we're able to energize 2 ATPs, and we're able to make 2 NADs, because that's basically the output of glycolysis. So they're going to do glycolysis just like we do. Except the next couple of steps are a little different. So let's do the lactic acid one. So what will happen is the pyruvates now will go ahead and be able to give a little more energy. So we're going to go ahead and get the NADs that came from this step. They're going to help the pyruvate a little bit, so they're going to lose a little bit of energy from that. And all we really end up with is the 2 lactates. So pyruvates get converted to lactate. And it's going to be done with this little bit of energy that's going to go on here. These hydrogen electrons are going to turn pyruvate into lactate, and this NAD will just jump over to here, get picked up with 2 hydrogen electrons. Next time it sees pyruvate, we'll turn it to lactate. If we go, and then that's why it's called lactic acid fermentation. So remember how you guys get this thing called lactic acid burn? It's basically when you don't have oxygen around, and your muscles will actually start to pile up a bunch of this lactate, and the lactate is an acid, so it starts to build up, it burns, and that's what that pain is coming from. It's just too much of this molecule starting to occur in your legs or arms or wherever. Let's go to the other side. Let's talk about the alcohol version. So we still have 2 pyruvates, and what happens with this one is the carbons get broken off. They leave as CO2 gas. The NADs, which got energized up here, are going to be able to convert this pyruvate into ethanol, and then the NAD is just going to go back up here, pick up 2 hydrogens and electrons, and then come back here to turn pyruvate back into ethanol. And you know that when you ferment alcohol, or even when you're making bread, you'll notice that one of the byproducts are going to be that gas, that CO2 bubbles, and that's what we're doing. Yeast is actually a prokaryote, but when you basically go ahead and take away its oxygen, that's why you kind of cover the starter material with tinfoil, or you cover it with plastic wrap so that it doesn't get a lot of oxygen. It's going to go into this process, which is called alcohol fermentation. The CO2 is where the bubbles come from, and then the ethanol is in the bread. So if you were actually to eat the bread raw at that state, you could potentially get drunk. There's way better ways of getting drunk. Don't do that. But what I would recommend is, this is also the way that they make beer and wine and things like that. They have to release some of the gas in the process of making beer and making wine, because that gas is this CO2 coming off, and then the ethanol is left behind. When you're making bread, you go ahead and take that loaf and you stick it in an oven, and the ethanol evaporates. So that's why you don't get drunk off of cooked bread. So the inputs are still glucose. The pyruvate gets reduced in both of these, and it just basically turns pyruvate into 2 other products, either lactate or ethanol. But what I'd like you to remember is that the products of fermentation are everywhere. You know about wine and cheese and bread, but you probably don't know about some other things that we can make from it, like yogurt and lasagna, and just you running is one of those processes of fermentation. What happens in your legs and your arms, it's just the lack of moving your legs around. So one last thing. So why not fermentation more often? The problem with fermentation is it's not very effective. Glycolysis makes 2 ATP, and that's it. And we make 36 ATP. So we're way more effective, we're way more efficient. We can use that glucose molecule and make a lot more from it. The other problem is it hurts. Lactic acid fermentation, when it gets into your muscles, starts to burn that acid, and if it's not dealt with pretty quickly, you actually could destroy muscle tissue. And it does this because it lowers the pH, it becomes really acidic, it eats away at muscles, and that's what muscle cramps are, is that muscles are starting to degenerate a little bit and come apart. They repair themselves overnight, but that lactic acid has to get converted into something else for your body to keep going, and that is going to happen in the liver. So can we get energy elsewhere? I don't want you to know this one for the exam, but what I wanted you to know is that we get energy from other sources too: proteins, lipids, carbs. It just depends on where that molecule sort of enters the respiration. Other molecules come in at different places, like some will come in just at the Krebs cycle, some will come in at the electron transport chain, and some will just come in at the prep cycle. But because I wanted to show you the complete cycle, that's why I showed you glucose, which actually does all these steps. And once ATP is made, it goes back and then gets rebuilt in the process. So proteins, lipid, carbs. It's kind of the circle of life. We bring in protein, lipids, carbs, and we sort of just break it down or build it up, and that's going to be called catabolism or anabolism. It's pretty awesome. And this is what I was alluding to before. We kind of discussed sugars, the glucose molecule, going through all the systems. So it'll go through glycolysis, it'll go through that prep stage that we talked about, it'll go through the Krebs cycle, and then finally it goes to the electron transport chain. But not everything does that. You can see some fats will actually jump right in because they're going to be 3 carbons. They're going to jump right in before the pyruvate step. You'll see some fats will look very similar to those 2 carbons already, so acetyl-CoA will just bring them in. With proteins, you'll notice that some of them will look just like pyruvate, some of them look like the 2 carbons with acetyl-CoA, and some will actually be able to come right into the Krebs cycle itself. So why I'm bringing this up, and please do not study for this part for the exam. I just wanted you to know, just for thoroughness, that you do get energy from other places.